if it gives like allow people to, usually it allows anyone to record.
Can you turn off the waiting room? Is that an option?
That's a good idea. How do we do that?
Let me try and reclaim host, and I'll do that real quick. Once you learn how to use Zoom, they change it on you. Anyone know where the waiting room options are?
I would think they'd be under participants. See here. And there's like more. But it also could be in the settings of Zoom itself.
Yeah, I'm in settings. I'm not seeing it. Yeah. Well, let me see here. Can you try and screen share again, Michael?
Nope, participant disabled.
Yeah. All right. Well, then I'm gonna have to just go ahead and give it back to you. And
Google says to turn off the waiting room, you go to tap security, or go to more and then tap security. Is that showing anything?
oh, okay. Thanks, Marianne. Tab security. Oh, I see. Enablement. Okay, got it. Okay, great. So that should work. All right. So I'm gonna hand it back to you, Michael.
Maybe since we want to get the maximum time, we start do introductions now and then give people a couple more minutes to.
That'd be great. Yeah. Yeah, wonderful. Okay, so let's go ahead and begin. So, Michael, thanks so much for joining us. I know you're a very busy guy, so really honored to have you here. And this is basically, you know, a UCSB centered group for the most part, surrounding largely general resonance theory, discussion groups, study groups, and speakers like yourself, but not entirely. We're kind of spread out now around the world. Colin Hales in Australia, some folks in Europe occasionally. So I'm a scholar with Jonathan in his lab in Santa Barbara, working for a long time on this approach to consciousness, and really intrigued by your approach to bioelectric fields in relation to consciousness and also biology more generally, which is my background. So Jonathan, please go ahead.
Yeah, so long time interest in consciousness. Much of my research has been on, well, it's really sort of all over the place, but mind wandering has been a long time research interest of mine. More recently, mindfulness, creativity, belief in free will, sort of philosophically related things, but also a long time interest in sort of philosophical perspectives. Very intrigued by panpsychism, by the role of resonance, by thinking about possible ways in which consciousness is manifested in physical reality.
Brian, go ahead.
I am a grad student in psychology at the University of Arizona. I work in SimiLab under Jay Sanguinetti and John Allen, and we do neuromodulation with ultrasound. And I'm interested in sort of trying to get at that big C consciousness. But I also have had a pet interest in bioelectricity and sort of the idea of like the biophysics of chi ever since I read The Bioelectric like a decade ago. So I'm really stoked to get to meet you virtually and hear what you have to present.
Jack, go ahead.
We can't hear you, Jack.
Sorry. I'm Jack Loomis. I'm a retired professor of psychological and brain sciences at UCSB. My specialties were in perception, spatial cognition, and virtual reality. and I have a longstanding interest in consciousness. And I saw your interview with Kurt Jamungal, and I was very impressed. So when I saw this opportunity to attend, I jumped at it. So it's so great to meet you.
Thank you.
Maria, go ahead.
Hi, I think I can echo some of what Brian said. So I just accepted yesterday my position to work as a postdoc in the Semilab. Yeah, I'm very excited. So I also work with ultrasound neurostimulation, but did my PhD in biomedical engineering. Definitely really interested in bioelectric fields or some of the bioelectricity stuff that's not really commonly talked about in the literature. I really have enjoyed reading some of your work, and I'm super excited to be here today.
Alan, go ahead.
You're muted, Ellen.
All right, great. Think I got that covered now. Yes, my name's Alan Macy. I'm the Research and Development Director at Biopac Systems. I am been involved with biomedical product design just for like 30 years, and very interested in biopotential recording and biopotential measurements of all kinds. And it's a total delight to be here.
Nikki, go ahead.
Hey, I'm Nikki Johnson. I joined MetaLab as a research affiliate in October, and my particular interest is in the transpersonal neural correlates. So what are the specific changes that take place in the brain, the body, and the spinal cord to trigger the subjective experience of enlightenment? So I've been working very closely with Tam and Jonathan and the team on GRT, but also some other projects, such as like James Elliott's online meditation studies, which kind of incorporate the belief in free will as well. Yeah, so anyway.
Amanda, go ahead.
Hello, so great to be here. I first heard you speak at the Diverse Intelligences Summer Institute this past summer, and really appreciated your talk on the continuum of agency from lower levels of consciousness to higher levels of consciousness. Really intrigued with how to depict kind of the interconnected relationships between levels of consciousness with an audiovisual experience that would provide a visual feedback for kind of what the direct experience of consciousness is. So as a person with a background in opera, using the sort of elements of music to metaphorically depict vibrational resonance theory and biorhythmic relationships, and those are the kinds of things I'm interested in. So thank you for being here.
Great, thanks everyone. So very quick introduction to Michael Levin. I'm sure all of you know who he is, but very briefly, distinguished professor of biology at Tufts, and has written, you've been active for what, 30 years now, Michael?
Something like
and
that.
really focusing on the bioelectric code in terms of evo-devo, regeneration, cancer, and then he's going to talk today about something that's not right in his wheelhouse, but is, I think, a mutual interest for all of us, is the relationship of bioelectric code to cognition and consciousness. And I've asked him to speak on these topics, maybe push himself a little bit more out of his comfort zone, but we'll see what he says. Thank you.
Okay, well, thank you so much. Really happy to have this opportunity to speak with you. It's an amazing group, a very different audience from the people that I usually speak to. So we'll see what you guys make of this. My background originally was computer science and artificial intelligence. My goal was I was interested in creating novel intelligences, and I shifted to biology, and so philosophy of mind and AI and so on. And I shifted to biology because I thought that there we would gain some new insight into what minds are and how we can construct novel ones and understand the ones that we have. And I think that's proven to be true. So I will share with you now what I think we've learned. Okay, so
Michael, just let me interrupt for a second. Can you record this?
I believe it's being recorded.
Oh, Perfect. And straight
Those
out.
are-
Sorry to interrupt. Perfect.
Yep. Yeah, I believe it's being recorded. Let's just check. Let's make sure of this.
I see it now.
It is. Yeah, it is. Okay. All right. Okay. So here's what I'm going to talk about today, which is something that that I now call kind of, it's a collection of ideas that I call technological approach to mind everywhere, or TAME. And what I'm going to describe today is, among other things, the idea that what bioelectricity does is it scales a basal cognition into a kind of collective intelligence. And in particular, I'm going to talk about the collective intelligence of morphogenesis or anatomical control. I'm going to tie that into a few other things. And if anybody wants to get hold of me afterwards, all of the contact info and the papers and the primary data and all of that are at these two websites. So the main points that I'd like to transmit are these for today. I take the view that the question of whether or whether a system is cognitive, intelligent, and so on is a bad question, and because it presupposes a binarization that I don't think exists. I think the real question for any given system is how much and what kind of intelligence, agency, and so on that system has. And I think we need to frame it as an engineering problem. It's not a question of philosophy, where some people will say, well, certainly, thermostats don't have any agency or whatever. And I don't think we can say that from an armchair. I think it has to be an engineering problem. And the way it's a scientific or an engineering problem is that I think we need to assign to any given system whatever degree of intelligence or agency gives you the best prediction and control over that system. Okay, so it's a little bit like Dennett's intentional stance in that way, but it's very practical, pragmatic approach. It's designed to drive forward research agenda, and what I'd like to do is to develop a framework that allows us to directly compare truly diverse intelligences, and you'll see what I mean by this. And I've developed a framework around the scale of the goals that any given agent can pursue, and the goals that are within that range define a sort of cognitive light cone of the kinds of things that agents can pursue and the kinds of goals that they can represent. And what I'm going to try to tell you is that morphogenesis is best understood not as a set of molecular mechanisms, but actually as an unconventional collective intelligence. It solves problems in morphospace, and an extension of the methods of neuroscience to bioelectricity outside the brain, or developmental bioelectricity, really provides an interesting new lens on this problem of how you integrate diverse competent subunits into one higher level cognitive structure. And I'm going to close with some interesting data from synthetic bioengineering in our lab, and then sort of talk about the future, which provides really an astronomically large option space for new kinds of bodies and minds that really are going to stretch our ability to use existing frameworks and terminology that's thrown around all the time, such as machine and robot and things like this, that I think are going to need to be completely revamped. So the goal of the framework that I'm working on is to enable comparison of truly diverse intelligences that that has nothing to do with their composition, meaning whether they're wet and biological or electronic or some other exotic material, or their origin story, meaning are they designed, evolved, or some combination of both. And so this means that we need to be able to compare and think about not only the familiar creatures that we're used to, but also some very weird ones like colonial organisms and swarms and so on, but also novel engineered life forms. And I'll show you some examples of these today. Of course, artificial intelligences that are being produced and are going to increasingly be produced, and at some point, perhaps even exobiological agents. At the very least, we should have an idea of how we would recognize one if we came across one. And the framework, the key to this framework is that it has to move experiment forward. It has to be empirical and has to drive novel experiments. So what I want to do is start off by trying to pull at the edges of typical model systems that people use and kind of highlight some novel aspects. and then move into what we've done in terms of this this technological approach, and then show you some novel organisms. Okay, so let's just start off by thinking about the typical example that people work with is some animal, and they say, well, here's my rat, and it has memory, and we know what the agent here is. It's the rat, and it has memories that it's formed, and it has preferences and so on. And one of the interesting things about biology is that it's actually extremely plastic, and this is going to be a theme throughout the talk. So here's a typical life cycle. You've got this soft-bodied creature, this caterpillar that crawls and chews plants. It's got a brain suitable for driving an organism of that type. Then it has to turn into an entirely different creature, which is hard-bodied and has to fly and drink nectar. Has a completely different brain. During this transition from here to there, the brain is actually largely liquefied. The connections are broken down. Many of the cells die. But what does remain actually is information, learned information. So things that the caterpillar learns are recovered in the behavior of the butterfly or moth. So this has very important implications for understanding what memory is in the first place. How do you store information in a medium that's completely malleable and changes on the fly? And it drives us to ask important questions, not only what's it like to be a caterpillar or a butterfly, but actually what's it like to be a caterpillar slowly changing into a butterfly? Because this is not some very long-term evolutionary process that we can sort of zoom in and just look at individual animals. This happens on the during the lifetime of a single cognitive agent. So we have to think about this plasticity. Of course, even every other animal and plant already has this issue because we all develop from one cell, right? And so there's this process of you start off as a collection of cells. Eventually, you're uncontroversially a cognitive agent. What happened in between, and how did that work? And even this in fact happens even more drastically in Planaria. So these are the champions of regeneration. That you can cut them into pieces, and every piece gives rise to a complete worm. But one of the things you can do is you can train them. So let's say you can train them on the fact that they're fed on this bumpy laser etched little surface, and then you can cut off their heads. They have a true brain, all the same neurotransmitters that you and I have. And the tail will sit there for 8 days doing nothing. And then eventually they will grow a new brain. And this now animal with this new brain remembers the original information. And so the rest of the body contains presumably contains some amount of that information, and it imprints it onto this novel brain as it's developing so that it can drive behavior. So this plasticity of memory moving from one place to another. And if we take memory to be some kind of indicator of individual identity, then one can ask, once you've chopped up a planarian into pieces, what happens to the original individual? It's sort of gone, but on the other hand, it's sort of now become 10 other individuals with similar memories. So all of the old philosophy of mind 101 kind of questions about duplicators and things like this are all perfectly doable in this creature. It actually happens. So what we're interested in is this idea of nested cognition, not simply the fact that the obvious fact that living things are a kind of a nested doll architecture, right? Where the swarm is made of individuals, which are made of organs, made of tissues, and so on. But actually, that every level here is not merely structural, but it's actually it solves problems in its own space. So molecular networks have kinds of memories and optimization that they can do. And collections of cells, tissues, organs. At every level, you have a kind of system with the ability to solve problems in its own special space. And because of that, one of the things we can do is we can now, and we'll come back to this, there are a number of themes that will sort of recur. One of the things we can do is in any multi-scale system like this, we can now swap out components. So within any organism, for example, at the cellular level, at the organ level, at the swarm level, you can replace its original evolved components with novel things that we have engineered. So you can put in things that are anywhere on this kind of plane that ranges from very passive materials all the way to sort of smart materials and smart kinds of artifacts, and they might be evolved and they might be designed, and there's an incredible interoperability in life, which allows you to change out the components on the fly. Everything works together. We already have humans that drive prosthetic limbs and wheelchairs with their minds. We have hybrids, which are robots with biological tissues driving them. We have all sorts of hybrids and chimeras, and then we'll talk more about this. And the interesting thing about being able to make all of these novel types of agents is that all of them will land somewhere on this continuum. This is Wiener and Rosenbluth's continuum of cognition. And you can see here that while there are phase transitions to more advanced capabilities, it isn't a binary line, right? There are many different types of simple to complex cognitive capacities. And so all of these things, which we've never seen before, will have to be placed somewhere by experiment. Will have to be placed somewhere on here, because we won't be able to assume, it won't be like it is now, where you can look at something and say, well, that kind of looks like a fish, and we know more or less where fish land. And so there you are. We're going to have things that we have absolutely no idea of their capacities, because they don't look like anything that we've ever seen before. We'll come back to that. So
let's a question there real quick.
Sure.
Could you go back one slide?
Yep.
So you mentioned phase transitions here in this framework. Do they suggest what the physical, the mechanism is behind that phase transition, or is this more conceptual phase transition?
Yes, I will make a couple I will suggest a couple of examples. They're not meant to be exhaustive. I'm sure there are many others, but I will show you one or two that I think are required for this.
Great.
Yeah. So okay. So let's take a look at this. So one thing that I would like to do is to generalize this issue of intelligence. And obviously there are many definitions of intelligence. I'm not claiming this is the only one, but it's one useful one. This idea of solving problems in arbitrary spaces is powerful, because we're used to things like this, where intelligence is a type of behavior, meaning movement in three-dimensional space. But actually, we are surrounded by intelligences that solve problems in gene expression space, in anatomical morphospace, which I'll show you many examples of today, and physiological space. The reason I think that we are very good at recognizing intelligence of this type is because all of our sensory organs point outwards. And so our training set of noticing other systems behaving is all about things moving in three-dimensional space. If we had senses that routinely examine the states of our inner organs, it would be really easy for us to recognize our liver, our pancreas, as fairly intelligent types of agents operating in physiological space and transcriptional space, solving problems. If we had access to our developmental stages, we would actually be able to recognize morphospace as well. But we traditionally don't have any of these. But I'm going to try to expand to that. And so we can define intelligence as the ability to navigate in these various spaces. I've just, for the purposes of the slide, knocked it down to 2 dimensions each, but these are very high, especially this one is a very high dimensional space. And one can also say that your amount of intelligence, or your degree of intelligence, is your competency to pursue goals in that state without getting trapped by local minima. So here, for example, there's the goal to which this animal wants to get to. In order to come around and actually get there, he would have to temporarily get further away from the goal. It's not a direct line to where he's going, but higher levels of organization allow a higher IQ for systems that can actually navigate to better maximum without getting trapped. And so your ability to do that is a kind of estimate of your intelligence. We'll get to another metric momentarily. But the nice thing about thinking of it in this way is that we can see that perhaps what evolution did was to pivot some of the same tricks in solving these problems. So very early life had to solve problems only in metabolic space, but then eventually genes came along and you had to solve problems in a transcriptional and physiological space. Eventually multicellularity meant you had to solve problems in morphospace. And eventually when nervous systems and muscles came along, you had to solve problems in three-dimensional space. And could it be that evolution makes use of some of the same principles to solve problems in these different spaces. Here's a transitional form. This is a slime mold known as Physarum. And in this system, and I'm going to play this movie momentarily, in this system, its behavior is its morphology. So what you're catching here by studying this organism is that pivot point of an organism where two of those spaces intersect, where learning to solve problems by changing your shape is in fact solving problems in three-dimensional behavior. And just to show you an example, this is something we discovered a couple of years ago. If you put the slime mold down here, you put one glass disc up here, you put 3 glass discs back here. There is no food on these discs. They're completely inert, just glass. The only thing it has to go on is mass. But as it turns out, it's able to, using a kind of sonar, a kind of sensing of pressure waves in the and tension in the environment, it's actually able to build a picture of the distribution of mass. And so what it will do is, for the first few hours, it sort of just grows out equally here. Okay, so here it just sort of grows. This is about 4 hours, and then at this point, it figures out where the bigger mass is, and that's where it goes. And this is quite reliable. It always prefers the bigger mass, and it always goes to the bigger mass first. So the behavior of this animal in solving this problem is in fact morphology. All it can do is change its body shape. By the way, this whole thing is one cell. Physarum is the whole slime mold is just one cell. So one way to start thinking about very unconventional intelligence is, you ask yourself, what do all intelligences have in common? If it's not what they're made of, and I don't think that's a good criterion at all, and if it's not how they got here, meaning evolution, design, whatever, the question is, what do they have in common? And I think one useful, again, not exclusively, not the only thing, but one useful invariant is their ability to pursue goals. And what you can then do is you can map out, and again, I've collapsed it into three dimensions of space here and then one of time. It's kind of like a Minkowski diagram in relativity. What you can do is you can sort of map out the size of the goals that any given agent can represent. Okay, this is not the size of their sensory reach or the size of their activity, but the size of the goals that they can represent. So if you're a very simple organism like a bacterium or a tick, you probably have very limited memory in the past, very limited predictive power forward, and all you're doing is measuring very local concentrations of things you care about, let's say sugar or butyrate or whatever. So your cognitive light cone is actually quite small. If you're another more sophisticated animal, you might have some considerable memory going backwards. You might have a little bit of predictive capacity going forwards. You will still never care about what happens in 2 weeks in the next town over. It's just impossible. Your cognition is not capable of holding on to goals of that magnitude. If you're a human, you might have an immense cognitive light cone. You may be working towards world peace and literally exerting efforts to manage the state of things on a planetary scale. You may be depressed because some of your goals are so far in the future they extend any plausible lifespan that you may have. Uniquely humans can have this problem. And if you're an alien or some sort of AI, you might have a weirdly shaped a cone that's bigger, smaller, or different than what we're used to. And of course, all of these are compound. So your cells, your organisms, and your tissues, and so on, will have their own different types of cognitive light cones. And the thing about so by focusing, there's a couple of interesting things that happen by focusing on the goal directedness of this. The first thing that happens is that it allows you to really compare and directly systems that are extremely different from each other. Right. The other thing that it does is it gives you some insight into a notion of stress. If we label stress as the driver between the target goal state where you'd like to be and where we are now, because in an important way, if you tell me what you're stressed out about, or the things that are capable of stressing you, I can pretty much guess where you are on this kind of continuum. So if the only thing you can be stressed about is the local concentration of glucose, you're one type of organism. If you're stressed about the future of the global stock market, you're a very different kind of agent, and anywhere in between. So one thing that we see then is that the scale of the goals on which you are working can shift during your lifetime. So here's one cell. This is a single-celled organism. It's called a Planaria. And this little guy handles all of its single-cell goals. There's no brain, there's no nervous system, but all of its anatomical, its physiological, its metabolic goals are all handled on a very small scale here. But when cells work together, they can work on very large goals. So here's one example: multicellular organisms like the salamander. If you were to amputate this limb, what the cells will do is they will immediately recognize that there's a problem, and they will work together, including the death of many of them, because programmed cell death is an important part of this. They will work together to build this, to rebuild this very large structure, this limb. They will stop working when their goal is complete. When does regeneration stop? When the correct structure is finished. They recognize, we'll get into this more soon, they recognize the structure is complete and the collective stops working, okay? Individual cells don't know anything about this. They don't know what a finger is. They don't know what the length of a hand is. Individual cells can only handle local goals, but collectives can handle very large goals. Unfortunately, that scale-up process, which we'll talk more about, can break down. So during the lifetime of this animal, one individual cell can become disconnected from the collective. And what happens then is that basically the goals on which it's working shrink back down to its unicellular past. And what you're seeing here is glioblastoma. These are cancer cells. The dream of each one of these cells is to become two cells and to go where life is good, and that is all. They are not going to be working on anything useful like an organ or a tissue. They will at best make a tumor, but basically they're just metastasizing as amoebas, and they treat the rest of the organism as just environment. So what we have to then keep an eye on, and this is where bioelectricity is going to come in momentarily, is that individual cells have an incentive. So an individual cell on the same scale has a little bit of predictive value, a little bit of memory, and a little bit of local quantities that it's trying to minimize or maximize, whatever. But surrounding yourself with copies of yourself in a multicellular setting has an important advantage. It gives you improved predictive value, because the thing that is the most predictive for you and the least surprising is yourself. And so this ties in nicely with Carl Friston's active inference framework or surprise minimization, and suggests that in attempts to predict and manage their environment, cells can get together and provide become a larger network, which has spatially a bigger area of measurement that it can take. It has more computational capacity for more prediction and more memory. And so what happens now is that whereas single cells can only measure and react to very small local things as part of remember what the goal is. The point of what it's trying to do is to take measurements of things it cares about, decide whether it needs to make adjustments towards the state it prefers, and then run that homeostatic cycle again and again to try to stay in a good area. Collections or networks, by virtue of connecting to each other, can measure, can represent, and can work towards larger goals. Their goals inflate. Okay, and we're gonna get into this in some detail. So the thing to keep in mind is that we have one example that a lot of folks have worked on over the centuries, which is in the brain. How do individual neurons tie together such that you get some sort of a central, the ability to pursue goals, let's say behavioral goals, that don't belong to any one individual neuron. And so this architecture won't surprise anyone. Here we have the hardware looks like this. We have cells connected to their neighbors by these electrical synapses known as gap junctions. They have ion channels on their surface that every cell sets a voltage, and they can communicate that voltage to their neighbors. Okay, and somehow this architecture is really, really key to binding individual cells into larger collectives that can pursue larger goals. Now, the interesting thing is, of course, that neurons did not arise out of nowhere. They didn't just suddenly discover all this stuff. Evolution found the system around the time of bacterial biofilms, and bacterial biofilms already use the same scheme to coordinate their activity. And as a result of that ancient origin, every cell in your body has ion channels. Most of them have these gap junctions to their neighbors. And this idea of using electricity to form networks is, of course, how we make computers, but evolution beat us to it by a long margin. And you can read more about that there. So just to give you a simple example, that type of model makes a simple prediction. The prediction is, could we in this kind of scenario, could we use the electrical connectivity specifically between cells and their neighbors as a marker and a control knob on the defection known as cancer? And so what happens is, when we inject it, as it turns out, when you inject a human oncogene into this frog embryo, eventually it'll make this tumor. But even before the tumor becomes apparent, you can already see, this is a voltage sensitive reporter dye where you can just get a map of all the electrical activity. You can already see that these cells are depolarized. That's the first thing that oncogenes do, is they cause an electric state that shuts down the gap junctions. They decouple from their neighbors, and you get this metastasis, basically this metastatic process, as the cells with no genetic with not necessarily any genetic damage, because you can cause this with lots of different mechanisms. They will now simply treat the rest of the animal as external environment. They'll crawl to wherever life is good. They'll reproduce as much as they can. They're on their own now. Cancer, from this perspective, is no more selfish than any other cell type. It's just that the self is now smaller. Okay, so what happens is that this in a multicellular body, and we'll talk momentarily about how this happens. The self is quite large because all of the cells are connected and they're working on a very large thing. As soon as you break that connection, your self is now tiny, and the rest of the animal might as well be external environment. And so the good news is that now, once we've understood that, you can now go back in and, let's say, misexpress an ion channel that forces the electrical state to be such that the gap junctions remain open. When you do this, here's the oncogene still blazingly expressed. This can be like a nasty thing like KRAS and so on. It's blazingly expressed, but there's no tumor. The cells are perfectly they behave perfectly normally. Now, if you were to sequence them, of course, you'd find the mutation. You'd say, you'd predict there'd be a tumor, and you'd be wrong, because the bioelectric state actually trumps the state of the hardware, trumps the genetic state. And you can suppress tumorigenesis this way, or in fact induce it by artificially breaking these connections. And so what's magic about the binding here is these gap junctions. And here's we'll take just a couple minutes to think about this. When you have two cells signaling to each other in a normal way, let's say there's some external ligand that comes along and hits a receptor. This cell is very clear on the fact that that signal came from the outside. Therefore, it can ignore it. It can do various things in response to it, but it knows for sure that signal came from the outside. Gap junctions are magic in the following way. When two cells are connected by gap junctions, each cell gets privileged direct access to the internal milieu of the other cell. That means that if something happens on this side, let's say there's some stress or injury that triggers a calcium flux, that calcium flux is going to propagate to its neighbor, and the calcium has no metadata on it that tells it where it came from. Once this cell experiences some second messenger, calcium or something else, it has no way to tell whether that's a memory belonging to this cell or to the neighbor. In other words, as far as this cell is concerned, it's a false memory because this cell wasn't the one that got stressed. But as far as this collective is concerned, it's absolutely a veridical memory. And the fact that you now, as a cell, you now here have engrams, memory molecules of traces of experience that you cannot distinguish whether they belong to you or to this other cell, that blurring of identity, who do the memories belong to, is what scales the system into a larger collective. It's the partial, and it's only partial, but it's the partial erasure of individuals' ability to keep track of which memories really belong to them, which binds this thing into a kind of larger scale cognitive whole that now operates on all the information. So the benefit you have is that you're now more clever, you have more computational capacity, you have more, you now have information you didn't have before and you couldn't have had before. But the downside is that you as an individual cell, in fact, either of these individual cells don't really exist anymore. There's now a collective which now consists of them both. And so you can just sort of scale that up and look at how information propagates through these networks in a way that erases the distinction between between one and another and makes a greater whole. So I'm going to show you some
question now,
please.
Yeah, so I guess a two-part question. First, do we know or have a rough idea of when gap junctions evolved biologically? And then second is, it sounds like you're creating a framework where any particular system of information or biophysical information pathways would lead to this compound individual creation in a larger sense of self. You're giving this as an illustration of how that might work. Is that right?
So yeah, so two things. Gap junctions are pretty ancient, but even before that, it doesn't actually have to be a gap junction. So I've pushed this idea into roboticists working in a completely different medium. It doesn't have to be an actual gap junction. It has a few features that are important to reproduce, which gap junctions meet. But even bacterial, I forget what they're called. There's some kind of bacterial thing where they get to sort of dock with each other that has the same property. It has to wipe ownership information on whatever molecules come through it, and it has to be able to be gated, being opened and closed. And even as far back as bacteria, before true gap junctions evolved, that those already existed. But gap junctions are themselves voltage sensitive, at least some are, which gives you an extra level of control, which is why it's so much better in metazoan organisms. As far as what does this mean for scaling, I think that yes, anything that meets some functional criteria would contribute to that scaling. It doesn't actually have to be a gap junction, but I think that's what evolution did. That's how evolution does it. Okay, so let's look at some specific examples. Now, one of the things I'm really interested in is morphogenesis and why. If we're into cognition, why are we talking about morphogenesis? What does that have to do with it? And so I want to address it starting over here. One of the things we'd like to do, and this is partly this way of getting into things, is what I use with colleagues and communities that aren't necessarily into cognition at all, but it's a way to try to make people care about this stuff, because I think it's very deep and fundamental. What everybody would like to have is an anatomical compiler. What you'd like to do is to be able to draw the animal or plant that you want, not at the level of genes or molecules or pathways, but you actually draw what you care about, which is anatomy, functional anatomy. And if we knew what we were doing, this piece of software would compile our drawing into a set of stimuli that would have to be given to cells to get them to build whatever it is that you want them to build. Now, why would we want this? If we had this, we could address almost every problem of medicine with the exception of infectious disease. So birth defects, regenerating organs after traumatic injury, reprogramming tumors, reversing aging, all of this would be possible if we knew how to tell cells what they should be building. Okay. Now it's very instructive to ask, why don't we have this? We are about as far away from this as you can get, despite all the amazing progress in genetics and biochemistry. So let's just ask a simple question. Here's an axolotl larva. These guys have legs. So baby axolotls have little legs. Frog larvae do not have legs. These tadpoles. We can and we have made chimeric embryos that are partly axolotl cells and partly frog cells. So we make these frogolotls, and now I ask a simple question: are frogolotls going to have legs? Now we have the axolotl genome, we have the frog genome. How come no one can tell me whether frogolotls will have legs? And part of it is because we really don't have at all a good understanding of how cellular collectives make decisions. We know the properties of the hardware of individual cells. We know what proteins they're going to have. We actually don't know how the collective makes decisions about what it is that it's going to build. And all of molecular medicine, so this is the chart of progress of computer science, where back in the day, in order to reprogram a computer, you had to physically change the hardware around. Can you imagine? You had to move wires. And nowadays, I say to my students, just be in constant awe that when you move from Microsoft Word to Excel, you don't get out your soldering iron and start changing the hardware, because that is what molecular medicine does. All of molecular medicine currently is focused down here. It's all about, we're gonna edit the genome, we're gonna change pathways, promoters, we're gonna change molecular networks. It's all down here. And what computer science shows us is that actually, if your hardware is good enough, and I think the biological hardware is definitely good enough, you can do something much more interesting. You don't have to rewire the hardware. You have to understand the software that binds the pieces into large-scale collective behavior, such as following an algorithm that you can then understand and manipulate. And so let's just look at how this works. Here's the cross section through a human torso. So look at this amazing invariant order, right? All of the tissues and organs, everything is in exactly the right place, next to each other, right size, the right shape. It all started life like this. And so a collection of blastomeres descending from a fertilized egg. And where did this pattern come from? Where did this order come from? And so people are tempted to say DNA, but we can read genomes now. And when you read a genome, you don't see any of this. What you see when you read a genome is protein sequences. You see the hardware that every single cell gets to have, but there's nothing directly in the genome about the size, the shape, the symmetry type. And so we really need to understand this process of how do cell collectives know what to make and when to stop. As workers in regenerative medicine, we'd like to know if a piece of this is missing, what do we communicate to the cells to get them to rebuild it? And as engineers, we'd like to ask, well, actually, how far can we go with this? Can we get the cells to make something completely different? Okay, can we get them to build something else with the same genome? So as I've already shown you, individual cells are very competent on their local scale. And the cool thing is that when they get together in multicellular organisms, that scaling that I was talking about allows them to represent, as a much larger network, goal states that are much bigger. And I'll show you a simple example of this. Here's a tadpole. Here's the brain, here's the gut, here are the eyes, here are the nostrils. In order for this tadpole to become a frog, it has to move the various organs around. So the eyes have to move forward, the jaws have to come out, everything has to move. And it was thought that, well, all tadpoles look the same and all frogs look the same. So as long as everything is a kind of a hardwired set of movements, you should get a normal frog from a normal tadpole. So what we did was we made so-called Picasso tadpoles. Okay, tadpoles where everything is in the wrong place. The eyes on the side of the head, the jaws are over here. Everything, everything is completely mixed up. And guess what? These things largely make pretty normal frogs because all of the different organs will continue to move in abnormal paths, sometimes doubling back if they go too far, until it gets to a correct frog face, and then things stop. So this is a system. It's not that the genetics somehow specifies these hardwired rearrangements. What in fact it gives you is a piece of hardware that can execute an error minimization scheme. It knows the correct set point, and it's able to keep deforming, even if it starts in an abnormal state. It keeps deforming until the error is as small as it can possibly get to. So what we have here is actually this down here is the standard story of developmental biology, and it's all focused on emergence. So you have gene regulatory networks, you have some physics, and eventually, if you do a lot of this in parallel, out will emerge some complex form. But what's really important about this is that there are actually feedback loops where when this set point, when the system gets away from the set point by injury, by disease, by mutation, by pathogens, whatever, teratogens, these feedback loops kick in and try to minimize that error. Now, on the one hand, now I have to say this is definitely not the standard story of developmental biology. On the one hand, biologists certainly know about feedback loops, right? But we're used to feedback loops that are single, very local quantities, like scalars, like pH, hunger level, things like that, temperature. Whereas what you have here, the set point of this homeostatic process, think of your thermostat, right? Your thermostat has this kind of feedback loop. The set point for this process is some sort of coarse-grained anatomical description of what a correct body looks like. And so people tend to not to like this because it reminds them of teleology, the idea that this is a goal-directed process. But fortunately, we've had, since cybernetics in the 40s, we've had a mature science of systems that are goal-directed objects, and they don't need to be, these artifacts don't need to be sort of second-order metacognition. You don't need to know you have goals in order to have goals. You just have to have this error minimization loop. And so this raises a number of questions which existed even before this was formalized, which is, how does it know when to stop regeneration? The most amazing thing about regeneration is that it stops when the right thing is finished. And how does it know what the correct pattern is? And so in our lab for years now, what we've been doing is following the strong and unusual prediction of this model, which is that we ought to be able to find the encoding, the pattern memory that tells these things what a correct shape is, and we ought to be able to learn to decode it, and we ought to be able to learn to rewrite it. And the thing about that scaling, the other thing about that scaling is that these memories, whereas individual cells, so living things are homeostats build upon homeostats, so that actually let's talk about this first. Individual cells all have a kind of homeostatic loop about things that individual cells care about, which might be a pH or hunger level or voltage or whatever these things are. And so they all execute these kind of loops, and they have this small the set points that they're using are all fairly small. So they're these small cognitive light cones. But when they connect in networks, what it allows the collective to do is to measure very large states, such as, for example, what's the distance between this bone and that bone, or what's the distance between the eyes, or in fact, how many eyes do I have? How many fingers do I have? Those kinds of things can be measured by coupled homeostats. So coupled systems can actually measure a very large state, and so that enables these large systems to work in more complicated problem spaces. And we already know that this happens. This happens in the brain, right? And so there you have these coupled electrical networks, and there's the resulting behavioral control software. And there's this the commitment of neuroscience is that, or at least of standard physicalist neuroscience, is that you ought to be able to read all the electrical states. You should be able to decode them, and eventually you have neural decoding. You will be able to recover the memories, the preferences, the goals, and so on of this agent. Well, because of this mapping, the fact that all cells are actually doing this, we have a very similar research program. Can we learn to decode the electrical activity of non-neural networks and extract the semantic content of this collective agent operating in morphospace, an anatomical space? And so I'll show you here the examples of what these pattern memories look like. Here's a frog embryo putting its face together. And so this is a time lapse, and then in grayscale, this is one frame out of that video, and you can already see this is voltage. This is a voltage sensitive fluorescent dye. Okay, so this is showing you in time lapse what all the voltages are, and you can already see the outlines of the face. This is a subtle scaffold that drives the downstream gene expression that then will drive the cell differentiation and cell movements that create the face. So here's where the animal's right eye is going to go. The left eye lags a little bit. Here's where the mouth, the placodes, everything is already sort of decided. And the reason I'm showing you this pattern, this example, is because in this case, the pattern memory actually looks like the thing it's supposed to represent. That's usually not the case. This is kind of a rare example that's easy to understand. If you mess with this electrical pattern, you can you will recapitulate the kind of birth defects that occur in humans and every other model system from mutations, for example, of various ion channel genes. If this pattern isn't correct, the face is not going to be correct. And so what we see here, this is a normal endogenous instructive bioelectric pattern memory that tells the cells how to make a frog face. Now, I've already shown you a pathological example of that in the cancer the cancer dye experiments. So what we did beyond being able to observe these things and characterize them is to basically steal a bunch of tools from neuroscience that enables us to rewrite these things in vivo. So all the same things that people do to work on synaptic plasticity or intrinsic plasticity, no applied electric fields, no electromagnetics, no magnetic component at all. This is molecular physiology to target the channels and gap junctions that are normally operating here. So we can control the gap junctions, meaning which cells talk to which other cells, or we can set directly the voltage by opening and closing ion channels. We can use optogenetics, we can use ion channel drugs, all the same stuff that neuroscientists use. When you do this, you can go in and do what in neuroscience is called inception of false memories, which people like Tonegawa at MIT have done. So if I go in here into this frog embryo and I inject a bunch of ion channel RNA encoding a particular ion channel to create an eye-specific electrical state in cells that are going to be gut. So these cells by default would have made a gut. But if I give them a pattern that says, no, make an eye here, what do the cells do? They make an eye. These eyes can have all the same layers, retina, optic nerve, lens, all the same layers. And there's actually a very cool phenomenon where if we only target a certain number of cells, the blue tells you which cells we actually targeted. What they will next do is recruit their neighbors, which we didn't directly target at all, to help them make the right shape and size of eye. So there's two instruction events. By the bioelectrical pattern, we instruct these cells that they're supposed to be making an eye, and then they instruct their neighbors to join with them and to execute this particular goal of making this lens. This is a lens sitting out in the flank of an embryo somewhere. And they instruct their neighbors. Using these kind of false pattern memories, we can induce fins, although tadpoles aren't supposed to have fins. We'll get to that momentarily. You can get otocysts or inner ears. You can get ectopic hearts. Here you can see that. You can get ectopic forebrain. You can get new legs or new limbs. And so you have some ability to respecify the large scale anatomical goal states on which these cells are going to work. Understand what we have not done is micro-specify cell types. So people will often say, oh, can you turn a stem cell into a particular type of cell? Maybe, but what this layer of control is supposed to be is not for that. It's not for micromanaging individual cell activities. It's for telling the whole large collective what it is that it should be working on. There are patterns that indicate different types of organs, and so this is not about individual cell behavior. This is about the instructive pattern of a large collective. Now let's look at how the memory works. And so this is in Planaria. We've been studying this. Planaria, of course, have this amazing property that you can cut them into pieces. The record is, I think, 275 or something like that. And every piece will regrow exactly what's missing, no more, no less. In fact, the other tissue will actually shrink so that the whole tiny little worm is in perfect proportion. They're so good at this, they're actually immortal. So there's no such thing as an old planarian. They live forever, basically. And all of the cells cannot simply be watching their local environment to know what to do, because if you cut an animal in half like this, the cells at the back end here need to make a tail. The cells at this end need to make a head, but they were exact neighbors until you separated them with a scalpel. They were at the same position inside the animal. So they can't simply know locally what, they have these radically different anatomical fates. They have to communicate with the rest of the fragment to say, well, what else is here? Do we have a head? Which way are we facing? What do we need to do? So that happens by virtue of a bioelectrical pre-pattern, this kind of voltage distribution that says to this fragment, one head, one tail. And in fact, if we go in and we depolarize this region to edit this memory to say, no, actually, you should have two heads, then this is what you get. The cells are perfectly happy to build to that shape. You can get two heads, you can get no heads. Now we can ask, I've been calling this thing a memory, and it's time to sort of cash that out. Why have I been doing that? Because if we take this two-headed animal and we ask a question, okay, we're going to remove the primary head. We're going to remove this crazy secondary head that formed. We're going to leave the nice normal middle gut fragment. There's nothing genomically wrong with it. We haven't touched the genome. This was all purely bioelectrically modified. We're going to let this middle fragment regenerate in plain water. The standard paradigm says, well, then of course you're going to get a one-headed worm because the ectopic tissue is gone. The genome is wild type. There's no drugs anywhere around anymore. It's plain water. So of course you're going to go back to your normal shape and make a one-headed worm. But the interesting thing is that if you look at the electrical circuit that actually determines this, it has two stable attractors, one at the single head state and one at the double head state. And so that's suspicious. Why would that be? Well, it turns out that if you were to do this, what you'd get is continuation of the two-headed phenotype. In perpetuity, as far as we can tell, these animals make two-headed worms. Now, the genome is identical. You're not going to get this information by sequencing their genome. The answer to how many heads is a planarian supposed to have is stored in the steady state of a real-time electrical circuit, just like volatile RAM, basically, that keeps this information, and we can shift it back. So we know how to now we know how to go from a two-headed animal back to a one-headed animal. And what I want to show you is that this is the beginnings of a really interesting, powerful. So this is the example. Somebody asked me at the beginning, what are the triggers of the phase transitions in that in Wiener and Rosenbluth's cognitive diagram? So here's one example. Take a look at this. This is a single-headed planarian right here. It has anatomically normal. It has gene expression of anterior genes in the anterior end, not in the posterior end. Has a bioelectrical gradient that says one head, one tail. And if you cut it, it makes a normal worm. Okay, so far so good. This animal, anatomically normal, again, gene expression normal. But what we've done is we've actually manipulated it such that it has an electrical pattern memory that says, no, two heads. Okay, now what happens when you cut this animal here is you get a two-headed animal. But really critically, this electrical pattern is not the electrical pattern of this guy. This is the electrical pattern of this guy. So a single body can hold at least one of two different ideas of what a correct planarian is supposed to look like. And it's a latent pattern memory. It doesn't do anything until you injure the animal. And at that point, the cells consult it and make what it says. But this is the beginnings of counterfactual memory. This is an electrical circuit that lets you remember things that are not true right now. This is the kind of the very basement of the ability to hold a counterfactual, to represent a counterfactual state. It represents something that is not true right now, because this is not the answer to how many heads do I have right now. This is the answer to how many heads am I supposed to have. And until you get injured, those two things don't really collide. But once you get injured, that becomes really that mismatch drives the morphogenesis. So I think
So where is that memory stored then? If it appears kind of spontaneously, where is it stored?
Sorry, I don't. So there's not. So nothing here appears spontaneously. This is the default electrical pattern that arises from the from the circuit comprising the ion channels that evolution has given us. It's a default attractor. What we can go in and do, because that circuit actually has this remarkable memory ability that it'll just remember when you set it to a different state, it keeps the new state. We can go in and force a two-headed pattern, and then it'll stick. It'll remember. It's like a flip-flop. There's a default. You hold it in a particular state, and now you can flip it from 0 to 1. This is exactly the same thing. It's a bioelectrical circuit that has the ability to reliably self-organize this pattern, but it also has the memory that if you change it, it'll keep the new pattern. And so that in the absence of injury, the cells basically ignore it. It's sort of a latent memory, but then it's recalled upon injury. So there it is. If you want to know where is the electric memory, you're looking at it the same way that if we were to take a flip-flop, the smallest piece of RAM that stores 1 bit of information, you would see. In fact, I have a cartoon somewhere that shows you how it works. It's exactly like this. If you were to just plot the voltage of that flip-flop, you would just see it low on one side, high on the other, or vice versa, depending on whether it was 0 or 1. And so what's really important now is this is a bridge between biophysical circuits that we study in the planarian and the kind of electrical computations that we see in neural networks that do really cool things like remember pieces of a pattern. So it's well understood in machine learning how it is that an artificial neural network can be trained on a pattern. Part of that pattern is deleted. The network is able to reproduce the whole thing. Okay, this is a fairly mature field that combines dynamical systems theory and these ideas in connectionist roots of intelligence. This is the link. This is how you go from how just physics. A lot of people say, well, that's not. I can see how the circuit works. That's not memory. That's just physics. This is how you go from just physics to the ability to store memory, to pattern completion, to counterfactual memories, first in morphospace, to have a memory of where you should be in morphospace, and later on, once muscles and nerves evolve, to pivot that same system to say, this is where I should be in 3-dimensional space, because I now have place cells and various other things that let me do pattern completion on things I see with my retinas. This is how that transition happens. And I think that's one important jump on that Wiener and Rosenbluth scale. Now, what's cool about all of this pattern memories is it's actually very deep. Not only can you get multiple different heads, but you can also get heads belonging to other species. So I can take this this triangle-headed planarian, I cut off the head, I perturb the electrical connectivity between these cells so they can't communicate with each other for about 48 hours. Then I withdraw the blocker, which happens to just be general anesthesia. They settle back down. When they settle back down, in some percentage of the cases, they find their correct attractor, but in many cases, they find other attractors belonging to other species of worm about 150 years, 150 million years distant. So roundheads like this S. mediterranea, or flatheads like this P. felina. And not only the head shape, but actually the shape of the brain and the distribution of the stem cells. So the morphospace is defined by the different potential attractors of this electrical circuit in the space. And when you, and normally it's very stable, the normal species specific head shape is very stable, but you can perturb the system, sort of raise in dynamical system speak, raise the temperature of the system, let it fall back down. And when it falls back down, it doesn't always land in the correct attractor, but the ratio, actually the frequency with which you get these animals is proportional to the genetic distance between them. So it's very reasonable to see how evolution sort of explores and canalizes in this space. You can go further and reach areas of morphospace that evolution does not utilize. You can make flatworms that are not flat at all. They're spiky, or they're cylindrical, or kind of combination of both. So there's a ton of things that perfectly standard wild type genetically identical cells can achieve if you modify the information content in the electrical circuit that's telling them what the collective is working on. And so, of course, this has all kinds of biomedical applications. This is how we learn to grow back legs on animals that normally don't grow them back, like frogs. We're able to induce this kind of regenerative response. We're now, and I have to do a disclosure here. David Kaplan and I are co-founders of a company called Morphoceuticals Inc., because we're now trying to induce regeneration in mammals and hopefully eventually in humans. So using these kind of wearable bioreactors. So just a couple more things I want to show you. We can skip some of these details unless people want to get to it later. But the idea is that we should be making a full stack model all the way from the genetic networks that set up the hardware of every cell, these ion channels, to the electrical dynamics, that the symmetry breaking and amplification dynamics that give you these bioelectrical patterns, and to the very large scale dynamics by which these patterns indicate organs, and ultimately to extract, to infer algorithmic kinds of descriptions of this, so that we can make changes here in a way that we can easily understand, not trying to micromanage at this level, which is basically hopeless in the long run. And so we're using machine learning to help us discover these kind of networks, and to discover interventions, to ask, how do I need to stimulate a particular circuit to get me a particular outcome? And one simple example of that is that, and so this is an example of fixing a really complicated birth defect. Here's a normal brain, forebrain, midbrain, hindbrain. Here's an animal with a genetic mutation. It works the same way with alcohol poisoning, fetal alcohol syndrome, and so on. This brain, the forebrain is gone. The midbrain and hindbrain are a bubble. If we go in, if we make a computational model of this electric circuit and ask it, what channels do I need to open or close to recover the correct memory from this state, it actually suggests a drug that already exists. In fact, it's human approved already, that targets a specific channel. And these animals get their brains back, despite the fact that they're still, the gene is still mutated, but they get their brain structure back, and they get their IQs back, their indistinguishable learning rates from controls. So what I want to get to at the end is this idea of coming back to this idea of biological goals. So we talked about this, the fact that in regeneration, it tries to get back to this particular state, depending on wherever you start. If you start here, it'll start here. If you amputate here, it'll just grow back from here on, and it gets to its correct state. In fact, it's the ability of cells to do this is actually quite remarkable. What you can do is, and this is very old work, this is a cross section of a kidney tubule in newts, normally made of 8 to 10 cells. What you can do is you can make polyploid newts which have giant cells. And as you increase the size of the cells, if fewer and fewer of them are needed to make the same lumen. If you make the cells truly immense, one cell will bend around itself and give you that same lumen. So different molecular mechanisms are being called up in the service of a large scale goal. So that's very interesting, and for people who want to think about top-down causation and so on, quite important. But the idea is that you can get to the same goal despite various perturbations along the way. These are serious perturbations, like the early embryo can be cut in half, and you don't get two half embryos, you get two perfectly normal monozygotic twins, and you get, or you can make the cells be much bigger. We don't have any technology where you can just change the size of all the components and everything works just as well, right? So this ability, this is very close to William James's definition of intelligence as being able to get to the same goal with various means, okay? And in fact, when we regenerate these frog legs, they don't grow back the same with the same path through morphospace as the development. They look more like plants. You get this single, the single stalk with a toenail, and then these off to the side, and eventually you get the same leg, but it's not the same path at all. So again, all of these systems are able to navigate morphospace in novel ways to get to where they're going, to the point where even functionally, if we take a tadpole and we remove the primary eyes and we put an extra eye on its tail, this eye connects to the spinal cord, doesn't connect up to the head. These animals can see perfectly well. They can learn. We have this machine that trains them for visual behavior. And so you can see the advantage for evolution is massive. Instead of evolving specific solutions to specific problems, which we evolve are a kind of multi-scale competency architecture that can, where all the parts are competent to get various things accomplished, and that allows you in their own space, and that allows you to solve problems you have not seen before. It means that this kind of robust goal directness, it buffers the negative consequences of mutations. Because if you have a mutation that's useful, but it puts the eye somewhere else, no problem. Things still work. You don't have a dead embryo, and it means that the higher level control circuits don't need to solve this immensely difficult high dimensional micromanagement problem. Instead, what they do is they bend the reward space for the subunits that are just pursuing their local thing. So this is at the very end. I know I'm already at an hour. I just want to show you the artificial organisms. What we wanted to do was to really ask about this idea of where do goals in these spaces come from? Okay, what is the actual origin? So what I've shown you are systems that can get to the same goal intelligently, but then there's a question of like, where do these goals come from? And you can see this right away, asking very, very simple questions like, what if I mix the stem cells from different kinds of planaria with round and flat heads? What are they going to make? Okay, we don't have any models yet that the field, despite all the progress on stem cell biology, there's no model that will make a prediction on what happens if you make a chimera of mixed. So what will be the anatomical goal state of an agent made up of these different types of cells? So what we did was, and this was in partnership with Josh Bongard's lab at the University of Vermont, and Doug Blackiston, who's a staff scientist in my group, led the biology. Sam Kriegman did all the computer science. We asked the simple question: can we make a completely novel organism from standard genetically normal cells, and ask what the goals of this organism are going to be, both anatomically and behaviorally? So what we did was we liberated some skin cells. These are just skin from the frog embryo. We didn't add anything, no transgenes, but we took away something. We took away the instructive interactions with the rest of the animal, and we just took some of these skin cells. We put them aside in this little, I'll show you again here, put them aside in this little depression. We just gave them a chance to reimagine their multicellularity. What are they going to do? They could have done a lot. They could have died. They could have made a flat monolayer. They could have crawled off and separated from each other. Instead, what they did was they made this little creature. We call it a xenobot because it's a biobot made of Xenopus laevis cells. It uses the little hairs that normally these cells use to redistribute mucus on the surface of the frog. It uses them to swim now, to row against the environment. And they can go in circles, they can sort of patrol back and forth. Here's some tracking data showing different behaviors of these guys. They can do a maze. This is a flat, there's no water movement in this. This is completely still water. There's no gradients, there's nothing, but you can see it navigate. I'm gonna show you this again. It's very interesting. So it moves forward, Now, at this point, without hitting the opposite side, it decides to take the corner. Okay. And at this point, something like proto free will kicks in, and for no reason that we know, just purely internally, it just turns around and says, forget it, there's nothing here for me. And it comes back to where it comes from. Okay. We have no idea what processes are going on. I mean, we're all obviously going to investigate this, but it's sort of like you're watching the very beginnings of self-directed motion. This is just skin. There's no brain, there's no nervous system. They self-repair. If you cut them in half, they basically try to seal up back to their original shape. Okay, like that. And if you express a calcium signaling reporter, you can see that, and then we glue two of them down so they don't move. You can see that it's very brain-like. This is exactly the kind of imaging you get when you do calcium imaging in, let's say, a zebrafish brain or a mouse brain. But again, there's no neurons. The skin. This is all being done by skin. And then the most skip that. The most amazing thing, which this paper just came out a week ago, is that being deprived of the normal ability to reproduce, which frogs do with sperm and egg and all that, these skin cells still figured out a way to make copies of their new organism, these new xenobots, which is kinematic self-replication. If you put them in a field of cells, they will run around, they will push these cells into piles, they will sort of corral them like this, like little sheepdogs. They'll just corral them, and they will assemble them into little blobs. Guess what these blobs become? These blobs become the next generation of xenobots, and they will do exactly the same thing, and they will go forward and make more bots out of. And so we can make, I think, up through 4 generations is so far we've seen. And so the same goal by different means. And the amazing thing is, so sometimes I've done versions of this talk where I show people these organisms. I don't tell them what it is. And then at the end I say, well, yeah, can you, somebody, Nikki, do you have a question? Sorry, you're muted.
I can hit it at the end.
Okay, great. So I'll say, what do you think this is? And people say, well, you got it out of the bottom of a pond somewhere. It's clearly some sort of organism. And I'll say, well, we got the genome. We sequenced the genome and say, oh, the genome, that's great. Now we'll get to the bottom of what this is. Well, guess what? The genome was 100% Xenopus laevis. Okay. So what's interesting about these creatures is that uniquely, as far as I know, uniquely on the face of the earth, these guys don't have a straightforward evolutionary backstory, because for every other animal, if you ask why do they have a particular shape or size or a color or what, behavior or whatever, the answer is the same, because eons of selection have shaped them to do specific things. These guys have never existed before. There was never any selection in the frog lineage to be a great xenobot. And so the fact that within 48 hours, these cells can self-assemble a coherent new organism with its own characteristic novel behavior and way of reproducing, and all these primitive, I mean, it's obviously a very primitive organism, but where did those come from? The selection pressure has always been to be a nice skin cell and sit on the surface of the tadpole and keep out the bacteria. So this is just the beginning, right? I think that what we're going to have, because of all these technologies, is the merger of evolved material, so different cell types from different creatures, design materials, so nanomaterials, microrobotics, and so on, and of course software in every possible combination. Hybrids, hybrids, cyborgs, everything that when Darwin looked out into the ecosystem and saw this variety, he calls it endless forms most beautiful. That will all be a small little corner of this enormous option space of novel creatures with new bodies and new minds, which are going to have collective goals and collective cognitive capacities that we will have to discover, because we're sure not going to be able to guess them by comparing them to anything. So this is it. This is the end. Just to summarize that, I think anatomical homeostasis, the ability to reach anatomical goals, is a really instructive kind of intelligence, which tells us something about different ways of scaling and different ways of solving problems. And it suggests all kinds of new biomedical approaches, which we are pursuing. One way to look at selves is via the boundaries of the kinds of goals they are capable of pursuing. And developmental bioelectricity is interesting because it's not just another piece of physics that you have to pay attention to in development. It is actually the medium the medium of memory and decision making for that collective intelligence of cells, that group of cells trying to achieve a particular outcome despite all kinds of perturbations. And now we can target these pattern memories to control behavior without micromanagement. And we're working on even higher level systems like training and just literally training tissues to specific outcomes the way you would train an animal. And so I will argue that evolution creates problem-solving agents, not specific solutions to specific problems. And it's a really great question for the future, which obviously we're going to try to address, is where does the ability to generalize come from? If evolution is supposed to be so short-sighted and reward immediate benefit, why is it producing things that are so good at generalizing to environments they've never seen before and novel solutions? I think that's a very deep puzzle. And synthetic bioengineering is going to enable a huge option space of new bodies and new minds, which is going to require us to really redefine a lot of terminology and a lot of concepts that we've had, and not to mention the new ethics that isn't based on what you're made of and how you got here. So lots more detail in these papers. I'm happy to, and many more papers, I'm happy to send out to anybody who wants. And I want to thank the students and postdocs who did all the work that I showed you today. Our funders, the animals, they do all the heavy lifting here. And that's it. I will thank you for listening and take questions.
Wow. Well, thanks so much, Michael. Really amazing stuff. A lot there. Let's go down the list here. Nikki, go ahead.
Okay. Yeah, thank you so much, Michael. That was really interesting. It was so cool to hear you talk about gap junctions, because I just ran across this last week in the context of the heart being described as a functional syncytium. Yeah. So a couple questions. It looks like gap junctions exist in various types of cells, and I assume they connect particular types of cells and therefore allow like the cells of a particular organ or system to be able to act as a single unit. Is that correct?
They don't necessarily, I mean, sometimes that's true, but they don't necessarily stop in between different, different tissue compartments. So you can certainly have different cell types connected by gap junctions, but the key thing about gap junctions is that they open and close post-translationally, meaning that they can be present, but they might be closed. And so tissues will both, and it's an either or. So both, if either side decides to close them, then they're closed. Both cells have to want to open them for them to remain open. So that means you can do all kinds of interesting and time dependent compartments and boundaries and things like this, right? Which is constantly evolving through the physiology.
Okay, and what prompts them to open versus close?
There are a variety of factors. There are things like intracellular pH will do it. Voltage is a really important one that will open and close. So think about what an amazing, so being both gap junctions and ion channels really are voltage gated current conductances. I mean, that's a transistor, right? That means you have historicity, you have memory. The fact that your current state of what you're doing now is a function of what you were doing moments ago allows you to have feedback loops, allows you to have memory, amplification. It's an incredibly powerful computational a computational tool. So I would say that voltage is probably the most important for us, but people study all kinds of things, phosphorylation and pH, and there's other things that control them.
Okay, another question. So in terms of the brain, I see references to astrocyte syncytium. So astrocytes being a particular type of glial cell. So is communication faster among those? Are you familiar with that term or?
Yeah, I mean, it definitely, right. The gap junction and other connections between glia and neurons are really critical. I think speed is, if we ask what's actually different about neurons than other cells, and we've had debates about this at various conferences where I'll say, well, what's a neuron? And so come on, here are 4 things that neurons do, and I say, well, every cell in the body does this. What's the actual difference? So one thing that is definitely different is the speed. So when motion in 3D space became an important thing, and animals started running around and trying to eat each other, that's when neurons optimized for speed, for solving problems really quickly in 3-dimensional space showed up. So direct connections, myelination, all this stuff. But other than that, if you sort of if you can take almost any neuroscience paper, do a word replace, and replace the milliseconds with hours, you'll have a perfectly reasonable developmental biology story. It's like they're faster, but a lot of the other stuff is the same.
Okay, interesting. And then finally, so gap junctions are like a physical connection between these cells, but at the electromagnetic level, like, is there shared resonance, or how does that work in terms of the intercellular communication?
Yeah, I'm not going to say anything about a magnetic component because I don't know. I mean, there are, ultra weak photons, and there probably is a magnetic thing that we don't know anything about, but I'm not going to say anything about that. Electrically, though, as soon as the gap junction is open, you have an electrical connection between the cells, and so you now have basically a partial sensation.
Okay.
And so gap junctions are really critical to turning individual cells into highly connected networks, where if I make a gap junction to my neighbor and we now short circuit the voltage difference, so we're at the same voltage, right? That's a very important computation that we've just done, right? We've erased an important difference between us, and that erasure of information is, I think, important for scaling.
Okay, and so the critical component is open versus closed in this like shared voltage then,
but in patterns, and that's why, I mean, gap junctions are also called electrical synapses. So everything that's true about the familiar chemical synapses is basically relevant here. So the same way that you can take a neural network and say, we're going to store memory in it by changing the state of the synapses via experiences, not by molecularly rewiring, but by experiences. That's exactly what happens with a gap junctionally coupled network. Your previous electrical state changes your open state, which changes your future electrical state, which now means you have a true synapse. You have a memory. It means literally a memory.
Okay,
Let's
cool.
go to
Thank you
Jonathan
so
now.
much.
You're muted, Jonathan.
Thank you, Michael. That was really, it's hard. I would say that your line of research, when combined, it's both philosophical and practical implications, is among the most astounding I've encountered. So
Thank you.
really, really impressive. So many questions. But I'll just try to ask just one or two. The one thing I'm wondering about is what you're into. Of course, you don't know, but I'm just wondering about your intuitions. What's your intuitions about consciousness as it relates to this? Do you think there's even anything remotely that it is like to be one of the bots that you have created? So I'll start with that.
Yeah, great question. I think that one thing that I can say for sure, which isn't much, but one thing I can say for sure is that in all of the things that we can track, I don't know how you, from a third person perspective, you can't really track actual consciousness, right? You can only track correlates, but every other thing that we can track, there is no sharp line to be biologically defensible anywhere. And so assuming that if we believe that the biology and the physics is somehow what underlies consciousness, I don't see that there's any way to put a hard line anywhere. So my strong intuition about this is that yes, there is something that it's like, and it's sort of minimal, and it's not anything like what it's like to be us, but it's something, and I see that consciousness going basically all the way down. Now, this and this is all, there's a paper, there's a very long paper that goes into all of this stuff in detail. That's sort of in revision now, so it'll be out hopefully soon. But long story short, if one question one can ask is, I'm very big on continuous. I think everything's a continuum. I don't think there's sharp lines anywhere here. But people do ask, well, is there a zero? Is there anything that's zero on this continuum, right? And I think that's motivated by this, by not wanting to say that rocks have hopes and dreams like we do, right? And that makes sense. And you don't, and it's fine, because when you scale down the organization, you have to remember to scale down the cognition. So of course they don't have hopes and dreams like we do, but let's ask, what does the absolute minimum of cognition and consciousness, what would that look, what would it have to have in order to be non-zero on the spectrum? So here's an idea, here's a proposal. I don't know if it's good or not, but here's a proposal. I can think of two things that I want to have if I'm not zero on the scale of cognition. One thing that I would like to have is some ability to be a goal-directed system, and minimally, extremely minimally. And all right, and the other thing I would like to have is I would like to have causes for my behavior that are not local. In other words, whatever I'm doing is not just because of the sum total of the forces on me right now. Because if that's what it is, then I'm a purely passive agent, right? Well, it turns out that even single particles meet both of those criteria, because you have least action principles that tell you that absolutely particles behave to optimize certain quantities like action. And quantum indeterminacy tells you that you can have cases where whatever you're doing is not determined by anything that's going on right now. Now it's random, which isn't great if you want to be a complex cognitive system. You don't want to be random, but for a minimal system, that's not a bad start. So as I think about what's the absolute smallest, sort of thing I would consider cognitive, I have to say that I think particles already meet that. So I would say, so based on that, I would say, no, there is no zero. There's a minimal form, and sometimes that gets washed out if you just clump them into a rock and it sort of goes nowhere and it just washed out and stays minimal. But biology does a lot better and it amplifies it. And when you start to scale up and amplify both of those things, indeterminacy and goal directedness, you get things that we can now start to recognize as, oh yeah, that does look like cognition. So that's my long-winded answer to that.
That's spectacular. Follow-up question, which you may have just answered, in which case I'll throw in another one which you can skip. It seemed as if this in the bots that they sort of in imperatives seem to sort of emerge that there was nothing sort of that before that led to. And so I'm wondering if you think, and perhaps that was your previous answer, there are some sort of fundamental imperatives that anything with this hint of cognition responds to. So are there, do you see fundamental imperatives? And then depending on your answer to that, if you have time, it vaguely remembers the phylogeny recapitulates ontogeny thing. And I've always wondered about that. And I'm wondering if your perspective informs or speaks to that sort of peculiarity of the way of embryonic development. Okay.
The great questions. I have to apologize. I just realized, and I know it's my own fault because I ran over, but I have to be on a student's thesis committee right now. I'm already late. Could we, but I really want to discuss this stuff with you guys. Is there any chance that we could reschedule the rest of this and just get on and chat? Is that a possibility?
Good. We've done 2 parts in the past with different speakers, so we'd love to have you back on and we can continue.
Yeah, that would be great. I'll come back. I'm really sorry. I did, it's my own fault, but I'd love to come back and chat about this. Okay, I just have to get on this student.
Right. We have many questions. Okay.
Please save them. I absolutely want to do this.
Okay.
Okay. Thank you so much.
Thank you so much.
Thank you so much.